Hello everybody, my name is Tanja Poljak and I am a medicinal chemist here in Fidelta. I got the opportunity to present one of the projects I worked on. This was a hit to lead campaign we did for one of our clients, eTherapeutics. Here is the outline of my presentation. So in the beginning I will tell you the aim of the project and the biology behind it. Then in brief, I will go through our client's network-driven drug discovery platform and the process of hit finding. And then in more details, I want to present you one of the chemotypes we explored here in Fidelta, the SAR around these compounds, Project Cascade, some synthesis, and eventually in vivo PK and PD results. So let us continue to the aim of the project. This project's aim was set by our client eTherapeutics. Our client recognized the gap in the current treatment of hedgehog-associated tumors and wanted to find new potent and selective hedgehog pathway inhibitors with a much reduced tendency to induce SMO resistance and with activity against tumors that already are resistant to the current treatment. If you haven't worked on the hedgehog signaling pathway before, this aim might be unclear to you. That's why I'm on my next slide. I will go briefly in the biology behind this project. So hedgehog signaling pathway is a well-explored cellular pathway in humans, and it plays an important role in the embryonic development and mature tissue repair in adults. Its uncontrolled activation also is linked with different kinds of tumors like basal cell carcinoma, medulloblastoma, and rhabdomyosarcoma. And here on the right side of the slide, you can see a simplified picture of the hedgehog signaling pathway from Nature Reviews Clinical Oncology. And as you can see, it consists of many proteins. The only two proteins I want to emphasize are first SMO or smoothant. This is a transmembrane protein and it is a known pharmaceutical target. There are already known inhibitors, really potent, that inhibit this protein. The only bad thing about this protein that it mutates easily and can cause tumor resistance. Another proteins I want to emphasize are GLIs, which are biomarkers related to the hedgehog-associated tumors. They are also biomarkers in clinics that predict the overall survival of the patients, and also we use them as a readout in our PD study. So this, as I said, the tumor resistance is detrimental for the treatment of the hedgehog-associated tumors, and this is the gap our client recognized. This was the initial idea that brought this project to life, as I will show you on my next slide. eTherapeutics apply network science to mimic disease biology. What they do then is they extract the network of protein interactions using measurements from human disease tissue and experimental data from their large proprietary database. After analysis, the software identifies the compounds that then predict to have the greatest impact on the network and hence the phenotype. The selected compounds are then tested in cellular assays that reflect the disease phenotype to confirm their activity. In Hedgehog project, our client got 1,146 compounds, which were selected in silico and tested in cellular phenotypic assay, which was set in Aurelia, UK-based life science CRO. This phenotypic assay was done on mice sarcoma-derived cell line, which were treated with the sonic hedgehog compound. This molecule is a ligand for hedgehog cellular pathway. Cells then, after the treatment, differentiate into osteogenic-like phenotype, and they start to express the enzyme alkaline phosphatase, which is a readout in this assay. Biologists in Aurelia also measured inhibition in biochemical assay against SMO and cytotoxicity of the compounds as well. At the end of this screening process, 63 hit compounds were identified, and with those 63 compounds, eTherapeutics came to us in Fidelta to do a hit-to-lead campaign. In first week, what we did here in phenotype, we looked here in Phi-delta, we looked at those 63 hits and clustered them into different chemotypes. We clustered them according to their chemical structure. We also looked for pains and potential toxicophores. We had checked chemical feasibility, and some initial IP space was also explored. Quite quickly, we started working, doing wet chemistry on the most promising chemotypes. And here in this table, you can see that this project had 2 phases. The first phase lasted for 15 months, where we did a broad chemical exploration on 4 different chemotypes. And there we identified the most leading, the lead chemotype, chemotype 1, which then went into the late stage, where we wanted to deliver compounds that would be appropriate for in vivo PD study. All this work was done in overall 21 months and in average with 4 medicinal chemists. In my presentation, I will focus on this lead chemotype 1 and show you results we got with those compounds. Here on this slide, it's a quite busy slide. It's a story around chemotype 1, which started with HIT compound that had cellular activity around 300 nanomolar, which is quite good. Also, it had some activity in SMO biochemical assay, but it was 10-fold less active in biochemical assay than in cellular. We looked at the calculated Phys-chem properties of the compound, and C-log-P was above 4. Which then we wanted to see the experimental value for the lipophilicity, and in-house we have validated experiment for determining chromolog-D. And after we did this experiment, the HIT compound had chromolog-D above 6, which means that this compound is quite lipophilic. Looking at the medchem indices, LE and LLE, here they're based on the cellular data. You see that lipophilicity ligand efficiency is quite low, which only confirms the statement that this compound is too lipophilic. And our first goal was to reduce this lipophilicity and more or less retain the same activity, which we did quite quickly with this compound ETXP1. You can see that the activity in cellular assay is comparable to the HIT compound, but the chromolog-D is over 4 units lower than the chromolog-D of HIT compound. And then we have the increase in LLE to 4.4. This is the structure of this compound ETXP1, and we did a broad chemical exploration around this compound, which eventually led us to different subchemotypes, which we also pursued each of them. Here, starting from the bottom of the compound, what we learned is that aromatic substituents are essential for the activity, and unsubstituted and differently substituted phenols were tolerated, as well as pyridines, but the best compound substituents here were aza-indoles and furopyridines. This central core piperidine and trans-cyclohexene were only tolerated. Sulfonamide was essential for the activity, and in this upper part, well, here azethidine, pyrrolidine, piperidine, and cis-cyclobutane were tolerated, and pyrazole as aromatic substituent. We could also introduce different functionalities there, like ureas, carbamates, amides, aliphatic and aromatic amines. So in this first part of the project, which lasted for 15 months, we also had a cascade set. We measured the cellular and biochemical assays in Aurelia, and here in Phi-delta we did in vitro ADME experiments. We checked for kinetic solubility, chromolog-D, stability in mice hepatocytes and liver microsomes, permeability in MDCK, and plasma protein binding. We also did some in vivo PK experiments to see and explore in vitro, in vivo PK correlation. Once we have reached better potencies and some idea about PK of our compounds, and on the other hand, we had in vivo PD model established in Crown, we went into a late stage of the project with more efficient screening cascade, which allowed us to select compounds from synthesis to tolerability study in 3 weeks. And this I want to show you on my next slide. So here is the late stage project screening cascade. As I said, we introduced this cascade to select compounds for in vivo PD model, which was established, and we had it scheduled for 3 compounds. And before this in vivo PD model, we needed to do tolerability studies for altogether 6 compounds. And from that study, we then selected 3 compounds to go into in vivo PD model. This tolerability study was done here in Fidelta. So what we did, as we synthesized new compounds, in the first week, in parallel, we screened the compounds against their activity and also did in vitro ADME experiments. We measured chromolog-D, kinetic solubility, stability in liver microsomes, permeability in MDCK and plasma protein binding. And by the Monday of next week, we got those results. And then good compounds were sent to full mouse PK. We had slots for 2 compounds per week to do both PEROS and IV. And on the beginning of the 3rd week, we would get those results. And if some compounds were okay, then we would scale them up to 250 mg and prepare them for the tolerability study. In the next 2 slides, I will show you 2 compounds that were selected through this cascade for the in vivo PD study. First compound is compound ETXP2, which had excellent activity in the cellular assay. PIC50 was around 8. The major advantage of this compound was its in vivo PK profile. So looking in mouse IV, it had low clearance, and also PEROS, it had high exposure and good oral bioavailability above 60. We wanted to test this compound in vivo PD model, and before that we sent it to the tolerability study, and it didn't show any clinical signs on mice. The dose for the PD study was selected according to the ppb adjusted potency and PEROS exposure at 10 mg per kg. And it was determined that this compound will go into in vivo PD study, dosed twice a day, 10 mg per kg. It was dosed orally. Another compound that we selected was compound ETXP3. It had cellular activity PIC50 around 7, so a little bit less than the first compound. The major advantage of this compound is then in SMO biochemical assay, its activity was above 10 micromolar, so it was selective over SMO. We've put this compound into in vivo PK, and although it had moderate clearance IV, PEROS profile wasn't that good, and the oral bioavailability was only 8. We nevertheless wanted to test this compound in the in vivo PD study, and we did the tolerability of this study IP at 3 doses. And according to those doses, we determined the dose of the compound that for the in vivo PD study. We also tested our compounds on vismodegib resistant cell line, and here in this graph you can see that in black are the IC50 curves of vismodegib on native cells and resistant cells, and you can see there is a drop in activity at least 20-fold. The curves for our compound are in orange, and you can see they're comparable for both native and SMO mutated cell line. So the activity of the compound wasn't affected by the SMO mutation. On the next slide, since I am organic chemist by training, I will briefly go into the synthesis of this representative compound ETXP3. So it's a 5-step synthesis. This is robust chemistry, and the starting materials are quite cheap. We start with 7-aza-indole and Boc-protected piperidinone in basic condensation to get the compound 1. After that, there is a reduction of this double bond and the cleavage of Boc-protecting group with trifluoroacetic acid. Next step is sulfonamidation and the alkylation of the pyrazole ring, which lead us to this compound ETXP3. And now we needed to do this synthesis in 1 week to get to the final compound to have the amounts available for the in vivo PD study, which we did. And here on the upper part of this slide, first you see the exposures of the compound ETXP3 in mouse dosed IP at free concentrations once a day. And you can see a nice dose proportional increase of blood concentration. And according to these exposures, we determined the dose for the compound ETXP3 in the in vivo PD study, which was 30 mg per kg per day, dosed once a day. Also, this compound ETXP3, when dosed IP on dose-free doses, didn't show any clinical signs in tolerability study. As I said before, we had an established in vivo PD model in Crown, and they ran an experiment on mice that had implemented human CALO6 lung cancer, and they measured after 5 days of treatment the expression of GLI protein. Crown used sonidegib as a control substance, and you can see that our compounds ETXP2 and ETXP3 are working comparable to the control compound, and you see the significant reduction of the GLI expression in comparison to the vehicle. They also took the healthy brain tissues from the mice and looked at the gene expression there. And for the compound ETXP3, they saw the reduction of GLI gene expression, which now we have brain target engagement with this compound ETXP3. This is a good hint of activity, since in the beginning I said that one of the major tumors that occur due to the uncontrolled activation of the Hedgehog signaling pathway are brain tumors, medulloblastoma. And to summarize this medicinal chemistry design, make, test, analyze cycle, so we started for this chemotype 1, we started with a HIT compound that had good activity in cellular assay, but it was too lipophilic. So the first idea was to reduce this lipophilicity, which we achieved with the compound ETXP1. See, there is a comparable activity in the cellular assay, but we reduced the lipophilicity to 1.7 chromlog-D. From this compound, we did broad chemical exploration and established different subchemotypes, which one led us to compound ETXP2 that had excellent activity in the cellular assay, and also it had good in vivo PK profile and the bioavailability of over 60%. It was active in the PD model. Another compound that we found was compound ETXP3 that worked in cellular assay and was SMO selective. So the activity observed in the SMO biochemical assay was less than 10 micromolar. It was tested also in the PD model and showed activity. And also we saw a brain exposure with this compound. And here is the whole summary of the project. So it started in eTherapeutics. They have recognized the gap in the available treatment of the Hedgehog-associated tumors and used their in silico network-driven drug discovery engine to select compounds that are then tested in relevant phenotypic assays in Aurelia. eTherapeutics got 63 compounds at HITs, and with those compounds, our client came to us to do the Hit to Lead campaign, where we worked. We were doing wet chemistry on 4 different chemotypes. One chemotype became the most promising, where we got orally bioavailable compounds with potency that were less affected by SMO mutations than in the vismodegib-resistant cell lines. We also achieved activity in the in vivo PD model, xenograft model set in Crown, and eventually 3 patent applications were filed around this chemotype. So I want to use this opportunity to thank eTherapeutics for giving us the confidence to work on this project with them. And also I'm happy that this collaboration ended with positive results. We achieved to get a proof of concept in vivo study, which is the best part of the medicinal chemistry project in this early stage. So thank you all for your kind attention and listening to this Hit to Lead story.